On the left side of this slide, for those of you near the front, you can see these dark circles that are healthy, living cells of the brain called neurons. We can induce their death by doing the sort of lesion I just described in the monkey. In the case of this growth factor called nerve growth factor, we can entirely prevent the death that is otherwise seen after the injury. And in Parkinson's disease, to repeat the slide I showed you before, these are the normal cells of this part of the brain that's vulnerable in Parkinson's disease, now shown in the monkey brain. We can model the death of these cells, and if we treat these animals with the growth factors, we entirely prevent the death of these cells. So, again, clinical trials moved forward, but they lacked an effective delivery method that was safe and delivered sufficient levels of the growth factor into the brain. So I'm going to talk to you about gene therapy as a way of delivering these growth factors into the brain. And the reason we use gene therapy is because direct pumping of growth factors into the brain causes intolerable side effects from too much spread of the growth factor throughout the brain, reflecting, in fact, the biological potency of these growth factors. If they spread beyond the areas of cell degeneration in Parkinson's disease, they stimulate healthy cells to have them become hyperfunctional and cause things like pain and abnormal sensations in the body. In addition, they cause cells to move from one area of the brain to another in response to the concentrations of growth factors. So we need another method, and there are basically two methods one can think of for delivering them to the brain. One is to pump the growth factor and limit its spread to one region of the brain. And this is the approach that a company called Amgen took in a recent Parkinson's disease growth factor trial, and they failed to see success with that approach. And that approach caused some toxicity to the brain. So another method is gene therapy. And what gene therapy basically means is that instead of pumping in the drug, the growth factor, we genetically alter the cells of the brain itself to make the growth factor. So you're not pumping in a drug, you're not pumping in a growth factor, you're genetically changing the cells of the person's brain or of the animal's brain to turn on production of the growth factor by inserting what's called the gene to make that protein into the cells of that person's or that animal's brain. So instead of delivering or pumping in a drug, we are changing the actual genetic makeup of cells in the brain to produce the growth factor, and we're doing it just in the area of the brain in which cells are degenerating in Parkinson's disease. So that's basically what gene therapy is. And when we do this gene therapy to deliver the growth factor in animal models of Parkinson's disease, this is what we see. And again, this is a small slideshow here, so you can't see it from the back perhaps, but we can model the death of these cells that are vulnerable in Parkinson's disease by giving a toxin to, in this case, the rat brain called 6-hydroxydopamine. Scientists recognize this as a standard way of trying to model cell degeneration of Parkinson's disease in rats. And when we give this toxin to the brains of rats, we lose the cells in this part of the brain called the substantia nigra. If we give gene therapy with the growth factor nurturin, or the other growth factor for the system called GDNF, you can see there is more dark staining in the brain here when we give the growth factor. We prevent the degeneration and death of these cells when we give the growth factor. And this shows the loss of these cells in rats that get the toxin. And this shows the preservation of these cells when we treat with either of these two growth factors delivered by gene therapy. And we can measure then how well the animals move after the toxin. And in animals that receive the toxin and no growth factor, they show an increase in abnormal movements. When they receive the growth factor gene therapy, they show a near absence of the abnormal movements that result from the destruction of the cells. We can also do this sort of testing in the monkey brain as we try to approach the human to have a better predictive possibility of making a judgment for moving to a human clinical trial. So in this case, once again, we've given a toxin to the monkey brain called MPTP. This toxin actually causes Parkinson's disease in humans, in a small subset of people who are drug abusers who mistakenly inject this as a substitute for heroin. This first appeared in this country 20 years ago. Heroin addicts were showing up in emergency rooms in San Francisco with Parkinson's disease. And when they were studied, it was found that they were injecting this toxin mistakenly. But this gave us our best animal model of Parkinson's disease. And when we injected the same toxin into monkeys or rats, they developed Parkinsonian features. And they show the loss of the cells in this characteristic part of the brain called the substantia nigra. So if we inject this toxin on the right side of the brain, then we cause the loss of staining of these cells that should normally be present. So this is the normal side of the brain, and this is what the brain looks like when you inject the toxin in the monkey. And if you treat the monkeys with the growth factor using gene delivery after giving the toxin, you preserve these cells, you prevent the degeneration and death of almost all of these cells. So again, the growth factor delivered by gene therapy shows potent effects in preventing cell degeneration. And again, these are the slides I've been showing you. This is the kind of effect you see at the level of individual cells of the growth factor delivered by gene therapy in preventing cell death. And then this graph shows functional improvement in monkeys that otherwise become Parkinsonian when they receive the toxin. So if we give monkeys a toxin and don't protect them with a growth factor, then they show scores on this motor scale that you see here. These are abnormal scores showing that they have Parkinsonian features. This is the equivalent, for those of you who are familiar with these scales, of the United Parkinson's Disease Rating Scale adapted for a monkey. And if we give the monkey the growth factor delivered by gene therapy, then up to 10 months after the toxin was delivered, they show nearly complete recovery from the functional abnormalities that otherwise result from the toxin. So in rats and monkeys, we can deliver the growth factor by gene therapy, we can prevent the death of the cells, and we can improve the functional state of the monkeys. So as a neurologist, when one sees these kind of data, you think, well, okay, that's not bad. That's the sort of thing we would like to try and develop as a human clinical trial. But again, the potency underlying this being the fact that if this worked in humans with Parkinson's disease, one would have the potential of preventing or slowing cell degeneration through the disease and prolonging the period of functional high quality of life. We can also measure the effects of this in another way in the monkey models. These are monkeys that have undergone what are called PET scans. This shows the activity of the part of the brain that's affected in Parkinson's disease. Normal activity is shown in the white splotch that you see here, and you've lost the activity on the right side of the brain as a result of the toxin. And when you give the monkey the growth factor delivered by gene therapy, you show a preservation of this activity. So the growth factor can protect the cells, it can protect the function, it can protect the metabolism. Again, this is a compelling body of data as a preclinical scientist to consider a clinical trial. So, there's a drawback to this approach of delivering the growth factor to the brain, of this gene therapy. The drawback is that once we genetically engineer the cells of the brain in animals or in humans, we can't turn it off. So it will continue producing the growth factor, as far as we can measure it, indefinitely. So if there's a problem from overexpressing the growth factor, how are you going to turn it off? And the answer is, we don't know how to turn it off. There's early development of ways to try and be able to regulate the gene therapy so you can adjust its levels. But today, as we move forward into the clinical trials, once we transfer the genes and do the gene therapy, we permanently genetically alter these cells of the brain, so that if a problem occurs, we won't be able to stop the thing that's causing the problem. Then you'd have to treat the symptoms that appear. So before moving to a human clinical trial with a form of treatment that we can't stop, you have to be very certain that the proposed treatment is safe for as long as you can practically look in a monkey model and in a rat model. So that was done. So the rat lives about 2 to 2.5 years. And the gene therapy was given to the rats throughout their adult lifespan, into aging. And over that time period, over that 2 to 2.5 years in the rats, there were no problems with gene delivery in the rat brain. And over 2 years in the monkey brain, with continued delivery of the growth factor over that time. It's a single treatment, by the way. So you do the gene therapy once. And that one treatment, that one session would hypothetically protect the would result in production of the growth factor for the rest of the life of that monkey. So over 2 years, there were no safety problems when doing this gene therapy, even when doses 100 times higher than proposed for a human clinical trial were done in the rat or the monkey. So if we give the rat 100 times more of the gene